So this is a work that came out of a very excellent collaboration during my employment at Johns Hopkins Hospital. I worked there for about 11 years, and I had a group of fantastic collaborators, scientists. The person who drove this project is essentially Dr. Rafael Guerrero. And he is a fellow who worked at Johns Hopkins who really wanted to understand what is the cause and what are the drivers of cancer in head and neck and human side as well. I know he had some mouse model systems, but we really wanted to study human head and neck cancer. And one of the best tools that he had at his disposal was sequencing. Fantastic technologies. Wonderful technologies on next generation sequencing or NGS as it's appropriately termed. And he used this methodology to really provide a more complicated picture than a cancer cell becoming genetically altered and then becoming cancer cells. It's kind of fascinating, and I think our work is concordant with a lot of other people's work, whether it be breast cancer, colon cancer, and that is that the microbiome within around the tumor, and always also microbiome outside of the cancer area, plays an important role in whether the cancer progresses or maybe better contained by their immune system. So it actually provides it's more of not just the tumor cells that undergo genetic changes and it becomes more aggressive with genetic changes. It's much more complicated than that. The cancer cells plays an intimate sort of a communal role with immune cells as well as the microbiome. And that we don't really know exactly yet in head and neck cancer whether this is pro-carcinogenic or anti-carcinogenic, but we know, I think the implication is that we have to account for the microbiome in head and neck cancer. Yeah, the challenge is we know that it's important for head and neck cancer, but we don't know the mechanism. And for this, we probably have to go back into the bench side. We need more scientists like Dr. Guerrero to actually query the important questions, go into mice or in vitro culture dish or other other tumor models to really tease out what is the role of the microbiome. Is it an offshoot of the presence of the cancer? And if that's the case, it becomes an excellent biomarker potentially that we can study, or do they play an important pathogenic role in the development and the progression of the cancer? Yeah, the future I would imagine is going back to the mice system, and the work basically showed that we have a few candidate flora that is highly enriched for advanced head and neck cancer. Namely, Fusobacterium is like 50-fold higher in the advanced tumors versus the early stage or normal tissue. So we know our patients and the samples derived from our head and neck cancer patients, they're telling us something. So what we need to do is go back and test whether Fusobacterium can have a promoting role in the mice system and prove this is the case. Oh, if Fusobacterium has a potential driver of oncogenesis, then these bacteriums can be controlled. We have an easy way to control them with antibiotics. So that has tremendous implication. And the other potential problem is that some of these bugs may grow out from excessive antibiotics use. So I can't see how we're going to change our antibiotics use, but we need to go back and go back into the experimental system and go do more of the wet bench work. Yeah. I just wanted to really score Dr. Rafael Guerrero, who really led the work. He was a leader in bringing all the team together. We had a team of geneticists, surgeons, as well as molecular biologists to really drive this project forward.